So this topic will kind of wrap up what we've done with the central dogma. We talked early about translation, then we got through transcription, and transcription left us at the stage where we had produced an RNA product, and then there's this further step, mostly found in eukaryotes, which is RNA processing, and has enough steps that I think it's worth talking about in its own presentation here. Among the steps in RNA processing is RNA splicing, and this molecule here showing the spliceosome, a rather complex molecule that holds the RNA transcript and cuts out pieces of intron. So just some framework for what we're talking about here. Again, just central dogma, looking at it from a different perspective. Double-stranded DNA, and one of those strands used as a template to make a single-stranded mRNA molecule. And the mRNA molecule gets translated into amino acids through the use of codons, as we've seen before. So this is all pretty familiar. And I wanted to put this up here because it also simplifies things down in a way that I think we take for granted what else is there. So as we'll see, not all of the DNA becomes RNA, and even not all of the initial RNA in eukaryotes goes on to become this polypeptide sequence. So there's a lot more DNA out there than this kind of diagram might lead you to believe. So if we zoom out a little bit and look at some more realistic data about genes and how they're arranged in the genome, you would notice there's a lot of spacing in between. And that's the point I wanted to make with this slide, is that of the DNA, not all of it ends up becoming mRNA. So mRNA, specifically this messenger RNA that is containing the codons for producing a protein, there's a lot more DNA there. So this is, the background here would be a DNA sequence, and this is portions of the sequence in these big blocks. These are the blocks that go on to become the final mRNA molecules. They have different directions to them. So as we saw with the promoters, promoter will determine what direction the gene is transcribed in. And then even among these big blocks of color here, so those are the disjointed portions of sequence that will become the final mRNA. Within that is also this pink shading, which tells us the coding region within that. So a small portion of the genes is actually that portion that contains codons. And as you see, a lot of their DNA space here is just space. So space in between genes is very common, and then space within a gene. So these would be the intron sequences that are important for keeping those genes together, but they're actually tossed out during the process of RNA processing. So again, this is mostly specific to eukaryotes, what we're talking about today. And one way to visualize the process of removing some of that extra RNA in between transcription and this final mRNA molecule, a lot of content is removed from the genes. So what we have here is the DNA, and they've skipped actually showing you the RNA that's transcribed from it, but the RNA transcript would look very much like the DNA sequence. So same length over this distance, and including the introns initially. And the introns are these regions that get taken out. So a gene, the transcript of a gene, can be broken down into introns and exons. And introns are the ones that get removed. I always find that a little bit, seems like it's kind of backwards for memorizing it. Introns, and one way to think about those is that they're in the way. And so they have to be removed before this final product can be used. That's just a mnemonic that I use. Maybe it's helpful for you. It's not really based on what the word means, but always seemed like it was backwards. The introns go out and the exons stay in, right? That's how it goes. So we have a couple of eukaryotic genes here, ovalbumin and cytochrome B, and those contain coding regions that will become the code for producing the protein, and also these spacers in between. So the coding regions, these shaded exon regions, they become part of the final mRNA molecule. So they get left in. So there are 8 of them in this molecule and 5 of them in this molecule. They get left in and stitched together to produce that final mRNA molecule. So the process of removing introns is called splicing. And this used to make a little more sense back when we had a little more of an analog media system. So if you ever maybe had an old version of music or a movie, music when I was a kid came on cassettes. You have tape, and movies came on VHS, and that was tape also. And they had this system, if you wanted to edit videos, you would have to edit by, if you had a long video, it would exist as this strip of magnetic tape. And if you wanted to cut out a scene you didn't like, you'd have to snip portions and tape them together. I didn't do any video editing in my younger years. Not as easy as it is for digital media. So basically, if you wanted to cut this part out, you'd have to physically cut it out and tape the remaining pieces together, because the message was a continuous strip of tape. And that's very similar to what we have with the production of genetic information. So we can have the initial RNA molecule, and then the final mRNA molecule. They're portions of sequence. This is perfectly good RNA nucleotide sequence, but it turns out that it's not really part of the message for making the protein. So it gets removed from the RNA so that the remaining pieces can make a coherent message. So it's a bit of a complicated process. On a molecular level, these 2 pieces of RNA on this linear molecule, they have to be cut twice to remove the intron. And then exon 1 has to be stitched up with exon 2. So it's important to cut, and then on a molecular level, not lose track of the 2 pieces that would need to be stitched back together. So for this class, I'm mostly interested that you know the concept, the fact of splicing, the terms of introns and exons, and we'll cover a little bit the signals involved. So there's a particular nucleotide sequence toward the 5' end of the intron, right at the edge there, and a particular nucleotide sequence at the 3' end of the intron. And those are the signals that cause this protein complex called the spliceosome to come in there and stitch things out. So this right-hand image here, not so important that you know the fine details. There's a lot going on, is kind of the point I want to convey here. And these proteins recognize and attach to the pieces, and this spliceosome molecule holds on to the 2 pieces, the exons that are going to need to be stitched together, and enables them to attach back together again. So there's a video I'll make available to you that you can watch that shows this process. For the most part, we just need to know where the introns are, the fact that there are some signals involved, and the results of taking out an intron. So this is one publication that highlighted some splice signals. So there are, as I said, toward the 5' end of the intron and toward the 3' end of the intron, sequences that determine where splicing is going to happen. So it's kind of interesting if you think about it. The exon has to contain the protein coding nucleotides, and so that kind of has its own sequence function. And you can see there's not a lot of conservation there. So could be any set of nucleotides, because those are part of the codons of the coding sequence. But right at this splice boundary inside of the intron, this is not going to do anything for the protein sequence, the protein coding sequence, but it is doing a very important job for determining that the splicing is going to happen there. So we have a couple of instances of a little bit of variation, a couple of major forms of these splice signals at the 5' end. Same thing over here, a couple major forms of the 3' splice signals. And then turns out there's this internal splice signal as well. All those are important for getting the molecule maneuvered in just the right way that it gets spliced out. This is a pretty conventional way to show a gene also. We've seen this a few times. The thicker blocks are the exons, and then these skinny single line pieces, those are the introns, meant to indicate that this has basically more information or vital information as far as making the protein, and then the in-between sequence, not so much as far as protein coding information or no protein coding information. So we see splice sites, and then also it's possible to have what they call self-splicing introns. Again, don't need to know all the details here, but this illustrates that it's not always the spliceosome that comes in and takes out the introns. There are actually some enzymatically active pieces of RNA. So depending how RNA sequences are put together, they can actually have a function. In some cases, the RNA folds on itself in such a way that it actually sort of cuts itself out. I don't know the fine details of that, but it's pretty awesome, I think. Just another, pretty much just two illustrations of the same process. This one highlighting the secondary structure of an intron. So a lot of these stem secondary structure formations, and then at some point in there, a cut. So we have the 5' end, 3' end, lining up in such a way that they will cut and remove themselves while also stitching the exons back together. Okay, so we're at this stage here, removing the introns. That's an important step, again, specific to eukaryotes there. So we've got our promoter causing transcription to happen. Transcription would happen through exon 1, intron 1, exon 2, intron 2, and so on, showing an end of transcription in this case. And then before the splicing, there are other steps that you've probably covered in other classes. I don't go into them in much detail, but I wanted to mention them in case they're familiar to you. And they'll come up just a little bit later. So a couple other modifications you get in eukaryotes are a modified cap at the 5' end. So 5' methylated G cap. And then at the 3' end, there's a little snip at this consensus sequence and the addition of what they call a poly-A tail. So that's just a many A nucleotides, just a run of A nucleotides at the 3' end. Those are part of the signal that this molecule is becoming one that is ready to be translated. So eukaryotes have a little bit of signaling that determines some kinds of RNA are different from others, and when they're finally prepped up and finalized, they have the molecular look of a molecule that needs to be translated. All right, so this is kind of the overall view of RNA processing. We've seen little bits of it. Here's the whole overview. So if you zoom out again and look at a gene, it's pretty easy these days to get a gene sequence if you know where to look. There are publicly available gene sequences. There are whole genome sequences. So you can get in there and find the sequence for a gene of interest without a lot of difficulty. So this is an illustration that your book provided, and it highlights some of the features. And I really like this way of thinking about things because it puts these nucleotide sequences in order, also in their space, and you can kind of walk through the process of converting a gene in the form of DNA, converting that into RNA, converting it into the final mRNA, and then finally into a protein. And I think once you've got that kind of understanding, you've got a really good handle on molecular genetics. So they've got some 5' ... Again, this would be one strand of the DNA molecule. And think for a second if this is the coding or non-coding strand of the DNA. So this would be the one we call the coding strand. Again, by convention, this is typically what you look at. Just keep in mind that the DNA molecule is double-stranded and would also have a complementary non-coding strand running in an opposite direction. Okay, so it's just sort of arbitrarily started at this point. And then later down, we get this highlighted region that has the TA, TA, and some other A or T nucleotides. That's the TATA box. And if you remember, that was a signal for one of the transcription factors to get involved. So that would be involved in starting transcription. And in this case, it's a little bit upstream from ... Here we go, transcription start site. So the position of the TATA box and other promoter elements determines where transcription starts. It doesn't matter terribly as far as the protein coding sequence, because somewhere downstream from the transcription start site is the start codon. So highlighted in darker green here are the regions that are the exons that contribute to the protein coding sequence. So ATG in the DNA is going to become AUG in the RNA, and that will start translation, as we saw in the translation content. So ... exon 1, and then at a point determined by the intron splice site, the exon stops, the intron starts, and this intron would get cut out. In this case, this is a short intron, and it's followed up by the next exon, and then another intron, and the other two introns, the one called intron 2 and intron 3, they've been trimmed down. So they actually have a big portion here. It says, we're not going to write all the sequence, but we left out 2,400 base pairs. Over here, same thing, intron 3, some was left out, 1,900 base pairs. So in this case, I think maybe kind of exceptional case, the introns are much larger than the exons of this gene. So there's a whole lot of transcription of RNA that's not going to end up in the final mRNA. All right, but we have exons 1, 2, 3, 4, and then the final exon ends with a stop codon. TGA is going to become UGA. That's a stop codon for the RNA. And that's upstream of the regions here that are part of the transcription termination sequence. So at some point, transcription would stop, and these signals, so this AAU, AAA, is a common sequence that helps to determine where this poly A sequence is going to go. So this is like the 3' modification of the final RNA molecule, also related a little bit to the end of transcription, so that termination sequence. So I think it's a little hard to appreciate this with lines of text that fold down. So I went and I found, actually did a look on the sequence here. It turns out this is a gene called IL-2, the interleukin 2. And I pulled up a canine version of this. So this is, sometimes you can get one of these, a map of the regions of DNA. So I don't remember what chromosome this is, but we're at position 17,750,000, 752,000, and so on. So someplace along this chromosome lies the IL-2 gene, and it's got similar illustrated parts as what we saw earlier. So thicker blocks become the exons, and then these single line pieces are introns, again, very large introns here, relative to the rather small exons. And then the difference between this darker, skinnier band here and the lighter, fatter band, the light fat bands are part of the protein coding region, and the darker parts are part of the final mRNA, but won't be actually coding for proteins. So we're going to think about this in space and time, as the cell would interpret the signals on the DNA molecule and then subsequently interpret the signals on the RNA molecules that are formed from it. So just going through time, we're going to think about these in terms of upstream and downstream. So as I mentioned before, this is a way to think about the order in time of things happening. Upstream happens before downstream, and then also a direction. So we're going to think about this whole gene moving left to right. The signals at the left happen first, and the signals at the right happen second, and third, and so on, kind of in order. So we're going to do blocks of sort of like a different pass. The first pass, we're going to transcribe. The second pass, we're going to modify the RNA. Third pass, we're going to do translation. So we've covered these concepts in a few different ways. This one, I really like this way of thinking about it because it ties it all together. So we're going to take our time walking through this one. So signals present on the DNA molecule would include the promoter and the terminator. So these are the signals that enable transcription to start. So I put the promoter pretty close to the transcription start site. That would be the first DNA nucleotide that is then transcribed into RNA. And then that RNA molecule is made from that point all the way to the terminator region, where that becomes the end of the RNA molecule. So promoter and terminator, they determine how long that first RNA molecule is going to be. So sometimes it's called pre-RNA, pre-mRNA. Sometimes I like this term, initial RNA, transcript. It's just the first thing that results, the first molecule that's made as a result of transcription. And in eukaryotes, this is not the final molecule, so it has to be transcribed and processed before it can be translated properly. So the transcript is simply a RNA copy of the DNA over the course of sequence from promoter to terminator. The second step, as we've seen, the second step we focused on, are these RNA splice sites. So contained within the DNA sequence are portions that are going to be coding for the protein, and those same portions get transferred onto the RNA molecule. And so what I'm highlighting here are the fact that the sequences are present on the DNA molecule, but I also want you to think about what molecule they're really interpreted on. So the splice site signals are interpreted on the RNA molecule, but they're a copy of what was present on the DNA molecule. So the sort of the signal was always there, but it's only interpreted when it gets onto the RNA molecule. Does that make sense? Let's hope so. Okay, so with the presence of those splice sites, those determine how splicing should happen. So I've drawn some dotted lines to the boundaries of those blocks. So the mature mRNA would contain only these shaded greenish areas, just stitched all together. And as we saw in that other illustration, the IL-2 gene has a lot of intron and relatively small amounts of exon. So when those are all stitched together, it's a lot smaller, but it is now streamlined to the sequence that's ready for translation. As we saw with translation, and something important to keep in mind with these, is that the start codon is not the first 3 nucleotides of the mRNA molecule. So the start codon, if you remember from translation, those were the first start codon downstream from the start of this molecule. So we went with the illustration here where the color changes in between the darker color and the lighter color. There is the start of the protein coding region, otherwise known as the start codon. So a little bit in from the start of this mRNA molecule. Translation happens between here and there. In this case, the stop codon is quite a bit upstream from the end of the mRNA molecule. So the rest of this is what they call an untranslated region. It's not going to get turned into any protein, but it's not the worst thing to have a little bit extra there. So as long as the sequence exists and is entire from the start to stop codon, then you'll get a functioning protein. All right, so lots going on here. There are signals and processes involved in transcription. There are signals and processes involved in RNA processing. And there are signals and processes involved in translation. And they all work together in this ordered fashion to get us to the end. Okay, so we've also talked about mutations and the topic of how DNA change can affect the protein coding portion of a gene. I thought I'd come back around to that topic now that we've seen a little bit more about the kinds of DNA sequence that can affect other parts of this process. So DNA sequence can be involved in the promoter region, the terminator region. We saw the splice sites and so on. So having a change in the DNA might be important to the coding region, or it might be important to some other process involved in central dogma or otherwise. So this is from your book, just an illustration of a single nucleotide change. And without knowing anything about the DNA, you wouldn't necessarily know what that change, what that mutation is going to do. But if you know what the DNA does or doesn't do, there could be plenty of non-functional DNA or DNA with no consequential function. In this case, this is say a recognition site for a transcription factor or something like that. Could be very important to have this change. So we talked earlier about mutations. What's the effect of a mutation? It always depends. So if it happens to be a piece of DNA that's important for a process, then changing it will have a major effect on that process. So back again to the OCA2 example. I think we're just about done running through these examples. I know you've seen a lot of them, but I mentioned the HERK2 gene, this gene that's upstream of the OCA2 gene. The specific SNP that has a major effect on eye color is located here inside of the HERK2 gene, but it's not actually part of a HERK2 exon. So it's inside of a HERK2 intron, and it's close enough to OCA2 that this is actually part of the upstream enhancer region of OCA2. So this is involved in making the OCA2 gene get transcribed. And that kind of shows us that we sometimes have actually some overlap between gene regions. So that was inside of an intron, kind of a little intron trivia for us. Another version of mutations, we looked at the consensus sequences for intron splice sites. These have their consensus sequences spelled out. The 5' splice site, this GT, is coming out to be very important. There's only G at the one site, only T at the next site. And so a change, a mutation in one of those sites can cause splicing not to happen there. And if you fail to splice out an intron, you might end up with that intron still contained in the final mRNA molecule. And then you'd be translating through essentially some gibberish. If the cell had to translate through an intron before it got to the next exon, that probably wouldn't make any sense. So major effect potentially on the structure of the protein that's encoded if the introns are not spliced out correctly. So GT, very important at this location. The 3' splice site, AG, very important at this location, in the context of this study at least. All right, so one more backtrack to the topic of translation. This kind of comes full circle for us. We saw these RNA modifications, so the intron splicing is important. And then we also, I just mentioned briefly, the 5' cap and the 3' poly A tail. These modifications are involved also in recruiting the ribosome. So in a eukaryotic gene, in order for translation to begin, all of this has to come together. So this cap binding protein refers to the 5' cap, and the poly A proteins are attaching to the poly A tail. So those structures on the RNA need to be there for translation to be successful. In bacteria, they don't have the modifications of 5' cap and poly A tail, but they do have something we've seen in a lot of contexts. They have this consensus sequence, so consensus nucleotide sequence of AGG, AGG. This is known as the Shine-Dalgarno or SD sequence. And that job of the SD sequence is to serve as a location where the ribosome can get attached. So the sequence there is recognized by the ribosome involved in translation, attaches to the RNA, and shortly after the SD sequence, usually there's a start codon right there, and then translation would happen. So a little note about the SD sequence is that according to this study here, it's not universal. So I think we have this, this programmed version of genes and cells. And the fact is that because of evolution, there's a little bit more randomness to the world, a little bit less precision sometimes when it comes to having exactly the same promoter or the exact same translation start signal in all genes. We got thousands of genes, even in a bacterial cell. And this is showing that not all of them, only about 2/3 of them, have this consensus strict AGG sequence. And it is kind of interesting, though, that the sequence AGG, this shows you a little bit more about what it does. It is complementary to a portion of the ribosome. So this is one of the ribosomal RNAs, and it has this piece right here, very important location, that is complementary, a little bit off here, A to U, G to C, G to C, A to U, G to C, G to C, complementary to the SD sequence. So it's actually, you might wonder why should a ribosome be made up of RNA? This is one of the reasons. So the RNA of the ribosome can form a complementary base pair interaction with the RNA of the SD sequence on the mRNA and start translation that way. Another thing we often see in bacteria are what we call polycistronic RNAs. So it's kind of a fancy word, but you can maybe remember the poly part of this refers to there being multiple genes next to each other. And the important part about polycistronic RNA is that all of these genes, protein 1, protein 2, protein 3 gene regions, they all get transcribed together. So one transcription event gets you 3 coding regions worth of RNA. And so actually the bacterial cells can translate through here and translate protein 1 and translate protein 2 and translate protein 3, all from this same RNA molecule. And I have on here advantages and disadvantages. One thing we'll see a little bit later in the course, we'll talk about gene expression in bacteria. And one advantage is that one of these polycistronic RNAs can cause, basically transcription will eventually cause the proteins to be made. So the cell can make proteins that have related function, produce them all kind of at the same time, or at least under the same conditions. And that's distinct from what we typically see in eukaryotes with these monocistronic versions that have one RNA produces one protein. So you probably thought about things a little bit more like this for most of the time, showing also the methylated G cap and the poly A tail. Again, if these signals are important, the poly A tail and the methylated cap, then those really, the start of this RNA molecule and the end of this RNA molecule kind of dictate that it's going to specifically translate just the one protein. But wait. So I love the position we're at in the stage of development and discovery. There's just always somebody upturning some old expectation. So I had taken it for granted for a long time that polycistronic RNAs were specific to bacteria. And here, just 3 studies coming out not too long in the past, showing that polycistronic genes are being discovered in eukaryotes across. We've got 3 different groups of eukaryotes. So mammals in the animal group, green algae in the plants group, and fungi in the fungi group, very distant organisms, all having some evidence of polycistronic genes. And just an example here, the blue 1, 2, 3, these are all pieces that make up genes, and they'd all be contained within the same RNA molecule. So still waiting for lots more evidence or new developments with this, really owing to the ability to get lots of new genomic DNA sequence now.